Hello everyone, welcome back to our overview of the heart. Now we are going to continue our discussion of the gross anatomy of the heart by taking a look on the actual inside, or the deep anatomy. Take a look inside the chambers, the valves, all of that. Okay? So again, we ended the last part of the lecture series by talking about the vasculature of the heart when we're talking about the superficial anatomy. So we're going to start our look at the detailed deep anatomy by looking at that same vasculature. So again, if you recall, arteries carry blood away from the heart, veins carry blood towards the heart. The two main veins that carry blood towards the heart from the systemic circuit are going to be your superior vena cava and your inferior vena cava, shown here. The superior vena cava and inferior vena cava are both going to dump into the right atrium. Okay? And we'll talk about that in just a little bit. Now again, arteries carry blood away from the heart. That blood can either be oxygenated or deoxygenated. The two main arteries of the heart are the aorta, shown here, and then the pulmonary trunk, shown here. Okay. The pulmonary trunk is going to split into the left and right pulmonary arteries, okay, which is going to carry blood away to the lungs where it will be oxygenated, and that blood will be returned via the pulmonary veins and empty into the left atrium. Now let's take a look as to the actual chambers of the heart. Again, if you remember, the right auricle overlays right where I drew that circle. But beneath that, that is where we have the right atrium. The left atrium, again, sitting slightly posteriorly and rotated laterally backwards as well, you have the left atrium. Beneath the atrium, or the atria, you have the ventricles. There is a right ventricle and there is a left ventricle. Now, a case in point that I do want to make out right now is that if you remember back to a slide about 10 slides ago where we talked about the myocardium and how I mentioned that thickness is proportional to workload. I want you guys to really think about this. The right ventricle is responsible for pumping to the lungs. The left ventricle is responsible for pumping to the entire systemic circuit, to the arms, to the legs, to the gut, to the head, to the neck, etc. It's responsible for pumping blood everywhere else. Now, if you think back to that hallmark of biology, that structure dictates function, think about the myocardial thickness needed to pump to the entire systemic circuit, to pump against all of the resistance in the peripheral vasculature, to pump blood all the way down to the feet and generate part of the pressure that brings it back. The left ventricle, or the left ventricular myocardium, is going to be significantly thicker than what you see in the right ventricular myocardium. Okay? Comparative size. I know this is just a rough estimate, but that's the right ventricle, and that'd be the left ventricle, comparatively speaking. The left ventricle has much thicker myocardium because it has to pump blood that much further and across that much more resistance. So just something to be aware of when you're asked to identify potentially a specimen or a section of the heart, either in an image or in the cadaver, what have you. Your biggest indicator as to what chamber you're going to be looking at and how to orient the heart properly is always going to be paying attention to that myocardial thickness, with again, the left ventricle having the thicker myocardium. Now, there is a specific type of muscle that lines each one of the two types of chambers. If you remember that the atria are sitting on top and the ventricles are sitting down here below, so that's the ventricles, and then you have the atria up here. Okay. The atria are lined with what is known as pectinate muscle. Pectinate muscle, this image makes it very kind of difficult to see. You can see it better in the cadaver, but you'll notice that pectinate muscle in here is very, very smooth. It's a very specialized type of muscle. Where on the flip side, trabeculae carneae, which is also a very specialized type of muscle that only lines the ventricles, there's a lot more meatiness to it. There's a lot more carnage. I like to think that, like I said, carnage, it's going every kind of which way. So if that helps you memorize it, go for it. If not, come up with something on your own. But again, the pectinate muscle is located superiorly in those atria, and the trabeculae carneae is located in the ventricles itself. The way I like to remember it too is your pecs are located superiorly or on your chest. The pectinate muscle are located in the pecs of the heart, if that helps. I don't know. Okay. But separating the atria, both right and left, you have a little segment of heart muscle or myocardium known as the interatrial septum. Separating the ventricles, both right ventricle and left ventricle, you have another similar structure right there, which is known as the interventricular septum. These names are very descriptive. They tell you what they do and what structure they run between. The interventricular septum, I also might add, is going to become very relevant when we talk about cardiovascular physiology in a later lecture series. Now, there are valves that also separate the chambers of the heart from the vessels and from one another. Separating the right atrium, separating the right atrium from the right ventricle, you have a valve right here. That is known as the right atrioventricular valve, also known as the tricuspid valve. Tricuspid because it has 3 cusps. I do want to point out right here and now that if you choose to memorize this, the right atrioventricular valve, you must spell that name out in its entirety. You can't abbreviate AV. I also will tell you why I almost prefer the tricuspid nomenclature here. I'll get to that in just a minute. Separating the left atrium from the left ventricle, you have another valve, which is known as the left atrioventricular valve, also known as the bicuspid or mitral valve. Bicuspid because it is comprised of 2 cusps. Mitral because the 2 cusps, if they're ballooned out, they look like a mitre. And a mitre in religious iconography can be most readily identified as the Pope's hat. So if you can picture that, that's kind of what this valve looks like. Now, the reason why I prefer the bicuspid and tricuspid valve nomenclature, because if you're trying to identify the flow of blood through the heart, if you remember that you try before you buy, you'll remember the correct order of blood flowing through the heart. Blood will flow from the systemic vasculature through that pulmonary circuit, which begins with the right side of the heart, before it gets to the left side of the heart and the bicuspid valve. So again, try it before you buy it. That's why I prefer bicuspid and tricuspid valve. You also have the semilunar valves. The semilunar valves are named such because if you look up here, the valves kind of look like half moon shapes, and there's 3 cusps in those half moon shapes. You have the pulmonary semilunar valve, which guards from the right ventricle into the pulmonary trunk. Guards from blood going out there. We'll talk about those valves in just a minute. And then you have the aortic semilunar valve, which guards from the left ventricle, left ventricle, going into the aorta. We'll talk about these valves here in just a second. So the heart works through contractions. You kind of already might have glossed onto that from when we were talking about at the very beginning of this lecture series. And we'll talk more about the actual contractility of the heart here in just a few lectures, and when we get into cardiovascular physiology. But the heart contracts, and the only way it's able to propel blood out through that contraction is by generating great amounts of pressure. So during diastole, when the heart is at rest, blood passively flows from the atria into the ventricles. When the heart begins to contract, it begins building up pressure in those ventricles. And that pressure in those ventricles is hopefully eventually going to squeeze blood either out that pulmonary semilunar valve or out that aortic semilunar valve. But if you recall, there's a nice open gap shut off by valves between the right and left ventricles and the right and left atria. How do we prevent blood from going back into the atria and redirect it out through either the pulmonary trunk or the aorta? Well, the atrioventricular valves will actually balloon up under pressure and close themselves off. But the pressure that they are under is so immense that they are actually anchored on their border to prevent them from prolapsing or allowing blood to blow back into the atria, either leading to a condition known as mitral valve regurgitation, which you might have heard, or heart murmur, which you also may have heard of. So these anchors, the cables themselves, are known as the chordae tendineae, or tendinous cords. Both names are applicable. On one end, like I said, they're anchored to the valves. On the other end, they are anchored to a series of structures known as papillary muscles. And these kind of look like mini Eiffel Towers that on one end, the feet are anchored to the endocardium. And then on the superior border, they are interlaced with the attachment points for these chordae tendineae. And what this does is, again, as the heart contracts and they start building pressure, it prevents those valves from ballooning up and prolapsing by catching the edges of those valves. If you can imagine my fingers being the edges of the valves attached to those chordae tendineae, and prevents them from blowing back. So because those cords are there, yeah, you'll get some strain put on those cords, but those valves won't blow open in reverse and allow the backflow of blood, because there you get a mixing of blood, and that's not conducive to normal circulation. I also want to point out that, again, papillary muscles are these Eiffel Tower-looking structures that anchor the chordae tendineae. They are also known as papillary bodies, and these, if you recall the special type of muscle that lines the ventricles, which is known as trabeculae carneae, papillary muscles or papillary bodies are just a very specialized form of that trabeculae carneae. So again, just some stuff you should be familiar with. Now again, these slides are for your reference. They go over kind of exactly what I was just talking about. So as blood passively fills the atria during diastole, which is when the heart is at rest, blood is going to drain down into the ventricles. When the heart begins contracting during its initial phase of systole, you get this phase known as isovolumetric contraction. Don't worry about that right now. We'll get to that in a few lectures. The pressure begins building, and blood is forced up against those atrioventricular valves and balloons those guys shut. Those chordae tendineae hold those in place, so that way the only way that blood can go out is through those semilunar valves. Once the pressure is great enough, those semilunar valves open up and blood flows out into the pulmonary trunk or out into the aorta. Now the semilunar valves work via gravity. Okay? After blood is ejected out during late systole, some of that blood is going to clear the aorta, some of it's going to clear the pulmonary trunk, but some of it's going to not make it all the way over those arches, some of it's going to fall back down. So if you recall, if some of it falls back down, some of it's going to land and pool in these cusps right in there. Okay? And that effectively is going to cause the valve edges to fold over one another, creating a nice occlusive seal and preventing blood from flowing back down into the ventricles. Now, the interesting thing is our body uses and capitalizes on this pooling of blood to actually supply the blood or the heart itself with the vital oxygen and nutrients that it needs. Okay? About 250 milliliters of, or about 5% of the blood that is ejected from the aorta is actually circulated this way down into the heart. So what happens is blood is pumped out of that aortic semilunar valve. Some of it is able to come up and clear that aorta, but some of it's going to fall back down and start pooling in these sinuses, okay? Or pooling in those cusps. And if you notice right here, there's two little outlets that lead into the coronary arteries. While you don't need to know the coronary arteries themselves, you do have to be aware of how the heart itself gets blood. So the coronary arteries just naturally act as drains and allow that blood to slowly percolate out and travel along its many branches. Okay? As you can see, the heart here is highly vascularized. So those arteries carry blood down and feed the ventricles, feed the atria, and then veins lining the surface of the heart. Artery being the pink fleshy one right there. I just drew the arrow over, and the veins being these larger blue structures right here. Okay? Those veins returning that blood back to the coronary sinus. So again, that is how the heart ultimately gets the blood. Now that we've finished our exploration of the deep anatomy of the heart, we'll reconvene here with a further discussion of the flow of blood through the heart, starting with one little erythrocyte returning from systemic circulation and ending with its ejection out into the aorta. Thank you for listening, and we'll see you here shortly.